Hello everyone.
Good morning, afternoon, and evening. So everyone, welcome to our today APMEN Tech Talk webinar. So I'm pleased to open the session on behalf of the APMEN. So as you may know, APMEN, short for the Asia Pacific Malaria Elimination Network, is a collaborative and sustainable regional network dedicated to advancing malaria elimination. The network brings together 22 national malaria programs and 54 partner institutions, connecting the country with relevant technical expertise and resources. Through our unique peer-to-peer collaborative approach, APMEN helps the country access the latest tools, evidence, and experience needed to address malaria challenges. Our work mainly focuses on strengthening capacity, exchanging knowledge, generating evidence, and supporting the country in adapting innovative strategy and tools. So this APMEN Tech Talk webinar series is designed to highlight our key technical topics identified by our working group, surveillance and response, vector control, and virus working group, as well as our interest groups, which include climate, environmental change, and malaria, border malaria, and autobiogen. So this session provides a platform to share the free updates, new research, the ratio guidance, and lesson learned from the country across the region. So today's webinar, we focus on the topic of growing urgency, so the intersection of climate change and malaria transmission. So as the climate continues to change around the world, it's becoming more important to understand how these changes affect malaria. So human activity also play a role in driving climate change and creating conditions that increase risk of malaria transmission. So this webinar will provide a platform for leading experts to share their insights and innovative approaches to using climate and environmental data to predict, manage, and control malaria outbreaks. So our excellent speaker will discuss various climate-related events that drive malaria transmission pattern. So I hope this session creates new ideas and strengthen our regional collaboration in the fight against malaria. I would like to hand over to Dr. Kimberly Fornes, so our chair for the Climate, Environmental Change and Malaria Interest Group, and also associate professor from the National University of Singapore. So over to you, Dr. Kimberly.
Excellent. Thank you, Dr. Phone, and thank you to everyone who joined today. So we have a really exciting lineup planned. If we can just go to the next slide, maybe. Excellent. So today we're going to speak a little bit about how we're actually addressing multiple hazards from climate and environmental changes and their impacts on malaria. I think one thing that we've found is that changes don't happen one at a time, but often we're addressing multiple compound hazards, either related to climate change or related to other factors such as control programs or urbanization or kind of wider changes. So this is a really kind of critical topic, and we'll hear from our three excellent speakers who will be speaking about different aspects of this in different countries within the Asia Pacific region. So next slide, please. So just to give an overview of the webinar format, the audio and the video will be disabled for the attendees, but please do continue to add your comments in the chat box. It's great to see so many people have attended, and feel free to introduce yourself. If you have any questions, if you can put these in the Q&A box, and we will address these afterwards. So we'll try and get to as many questions as possible from our three distinguished speakers. So we will have the three presentations, which we'll do back to back, followed by a panel discussion and some of these Q&As. And at the end, we'd be very grateful if you could stay for a quick poll. Next slide, please. So just to introduce you to the APMEN Climate, Environmental Change and Malaria Special Interest Group. We launched this last year, but we're increasingly thinking about this space. So how do we address these challenges of climate and environmental changes on malaria transmission in our region? And to do this, we're asking some key questions. So what are the impacts of climate and other environmental changes on malaria in the Asia Pacific region? And what is the evidence we have so far? Critically, how can malaria control and elimination programs adapt to these changing environments? And what types of strategies can we use to mitigate these impacts of environmental change? And finally, we're kind of really thinking about how we run more sustainable programs for ourselves as well. So we have a few kind of key interest groups, key activities that we will be conducting. We've been looking at kind of reviewing the research which is available on the impacts of climate and environmental changes on malaria within this region specifically. From this research, we're thinking about how we can actually support the development of policy recommendations for governments addressing the challenges of climate change. And really critically, what we are doing right now is really thinking about how we facilitate knowledge exchange among researchers, public health practitioners, and also climate and environmental scientists. So we're very grateful that everyone has joined, and a particular thanks to our speakers who are adding their expertise to this. We're very keen to hear from you about what you think would be helpful. So if you do have any suggestions or you have a topic that you'd like to see explored or some research that you would like to share, please do email me. My email is here, but you can get in touch with any of us here at APMEN. So excellent. So without further ado, I'll introduce our first speaker, Dr. Tung Win Yoi. So Dr. Tung is actually our climate scientist speaking today. He obtained his PhD in ocean and atmosphere and climate from the University of Toulouse in France in 2022. Currently, he's working as a postdoctoral researcher within the DART project in the Oxford University Clinical Research Unit in Vietnam. And his research interests are in atmospheric and oceanic modeling, climate extreme events, climate change, and adaptation. So we're very excited to hear from you, and thank you again for making time.
Okay. So hello everyone. I'm Tung. So actually I'm a climate scientist. So it's a very honor of me to present my work today. So I will share my screen. Oh no. So sorry, I will need to reopen the app because if we need to reopen to share the screen. Sorry, I will come back in 1 minute.
Excellent. I hope it works. So we'll just give 1 minute, the joys of Zoom, which I think we all can relate to.
Okay,
Great.
so sorry for the delay.
No problem. I think we've all been there.
Okay. So today I will talk about the identification and trend analysis of the compound meteorological hazards along Vietnamese coastline. In Vietnam, we have a very long coastal line, which is like more than 3,000 kilometers. So it is a country that has been strongly affected by the climate change. Therefore, understanding the characteristics of different hazard types is crucial. So for some extreme events like the hot temperature, the heavy rainfall, and the tropical cyclone that can be considered as a meteorological hazard, and these hazards can be destructive and have negative impact on various aspects of life. For example, you can see here in this figure, it is a flood induced by the heavy rainfall in Vietnam, and here is the drought also created by the missing of the rainfall. So as I said before, the three type of hazard can have negative impact to life. For example, the heat wave can decrease the labor productivity and reduce the durability of the building material. On the other hand, the heavy rainfall can trigger another type of hazard, for example, floods and landslides, and also it can increase the infectious disease case. For example, when heavy rainfall occur, it can move, it can clear the breeding site of the mosquito, but also it also create new breeding site for it. So at a time of very rainfall, we may have a decrease of the disease created by the mosquito, but then after like some weeks, we can have an increase of the infected disease case. The tropical cyclones can be also have multiple impacts due to the heavy rainfall, the strong wind, and also the storm surge, especially in Vietnam, where we have a very long coastal line. And also it can trigger other hazards like flood and landslide due to the rainfall in the cyclone-induced rainfall. Uh, Though we have several types of the hazard, the study of the compound hazard in Vietnam is remain limited. So in this study, we aim to conduct a case study for the change of the compound hazard in Vietnam that consider the extreme rainfall, the extreme temperature, and the tropical cyclones along the coast of Vietnam. In this figure, you can see here it is a location of the 10 meteorological hazards that I will use data for the analysis. In this bracket, you see it is a number, the percentage of the missing data. So we see that most of it have less than 1% of the missing data from 1980 to 2018. So which is nearly 40 years. And here in this map, you can see the track of the disease that affect Vietnam in the same period. Yeah. And the study at my study at period is from 1980 to 2018 that I just said before. The temperature hazard is defined as the day which has the temperature higher than 95 percentile of the all of the period. It's the same for the precipitation, where the cyclone hazard is defined as a point which is located within the radius of 250 kilometers from the cyclone center. And a compound hazard here, it is defined when ending time of a hazard event and the starting time of another hazard is less than 10 days, for example, here. For example, here in this figure, you can see CE for the cyclone, T for temperature, and R for the rain. So if we have a cyclone here in this day and a rain hazard on this day, this one is considered as a compound hazard. And here is a result. In this figure, you can see the number of compound event per year in all of the 10 stations within the threshold of 7, 10, and 15 days. And what we see here is that in almost all of the stations here, we see an increasing trend of the number of compound hazard per year in all of the stations here. 3 out of 10 stations show a significant trend of the increasing in the compound hazard. And then if we look at the number of tropical cyclone per year and number of the rainfall hazard per year, we see that the trend of these events is not significant. We can have some station with increasing trend, for example, here station 5, but we also have some station with a decreasing trend, for example, station 7 here and station 8 here, but they all are insignificant. However, if you look at the number of temperature hazard event per year, we will see that they have an increasing trend in almost all stations. I think that is the 7 out of 10. And also the station which have the significant increasing trend in the compound hazard is also the same stations that have the significant increasing trend in the temperature hazard. So it means that the temperature hazard here is the main factor that drives the number of compound hazard increasing along the Vietnamese coastline. Also, if we look at the seasonal cycle of the compound hazard, we see that here in this figure, you can see this is a station from 1 to 9, under 10, and here is the time from January to December. We see that from in the north, the Campa hazard tend to occur later from June to August. Why? When we go down to the south, the Campa hazard can occur sooner from April to May. So it means that if we want to build a mitigation plan for different places, we need to consider it in a different time frame. Yeah. And finally, in this figure, you can see the pie chart for the percentage of the type of the component that is linked to the Campa hazard. For example, as you can see here, the orange one is temperature, the blue is for the rain, and the green is for the cyclone. So you see that in the first half of the year, the temperature is a factor that contribute most for the occurrence of the Campa hazard. While for the end of the year, for example, from September to December here, the Campa hazard is mostly contributed from the rain and the cyclone. So in summary, in Vietnam, along the coast, the Campa hazard typically occur from March to November. However, in the early summer, the temperature hazard is the main contributing factor, while in the later of the year, the rainfall and the tropical cyclone hazard becomes dominant. Also, we see that increasing trend in the number of Campa hazard is observed in most of the stations along the coast. Due to we see that the trend of the temperature hazard as a primary contributing factor to the trend of the Campa hazard. Therefore, with the global warming and with the increase of the temperature hazard, so we tend to have the increase of the Campa hazard in the future due to the increase of the temperature hazard event. For the future work, as we can obtain more data, we can include more comprehensive list of the Campa events, and also we plan to assess the risk posed by the Campa hazard for better adaptation and mitigation plans. And this study has been published in the Natural Hazard. And if you would like to have more information, you can refer to this paper. So that's all for my presentation. Thank you.
Excellent. Thank you so much, Dr. Tung. I think it's really important to start thinking about these patterns where we have these compound hazards, particularly when we think about malaria control. Often we are only thinking about some of the determinants of transmission, like rainfall and temperature, but we know as well that some of these other factors really even just impact our ability to deliver control. So whether we can access an area, the health of the workers. So I see some questions below. Please do add any questions in the Q&A. So we will have questions all together at the end. But thank you again for your talk. So our next speaker will be Dr. Alex APMEN. So Dr. Alex is joining us from the School of Public Health at the ICMR, National Institute of Epidemiology in Chennai, India. He has a wealth of experience on malaria and has been working on vector-borne diseases since 1988 on topics including malaria, dengue, and chikungunya, from projects ranging to the operational feasibility of bioenvironmental control of malaria vectors, the first use of GIS systems to actually inform the malaria surveillance system, as well as innovative strategies of control. So Dr. APMEN has been involved in high-level investigations for analysis of post-flood scenarios and has done some really exciting work on microclimate and urbanization. So we're very grateful to hear from you today. So over to you, Dr. Alex.
Good morning, good afternoon to all. Just want to make a small correction. So I'm a scientist at ICMR, National Institute of Malaria Research, though I'm stationed in National Institute of Epidemiology. I'm a scientist from National Institute of Malaria Research. Okay, the talk which I'm going to give is climate change, basically on microclimate studies, which we have taken up in Chennai, in India, and urbanization and the vectors which are involved in as far as urban malaria is concerned. Can we have the next one, please? Next slide, please. Yeah. So the progress in malaria control as far as India is concerned. So if you see the 6 decadal malaria trend, you could see the cases, the blue bar represents Vivax and the red one falciparum. Altogether, it's total cases. The primary axis on the y-axis shows the number of cases. And then you have the percentage of Plasmodium vivax and falciparum, which you can see dipping, and you could see the cases of vivax and falciparum were about 50% each when around 2011 and 12. And then ever since there are changes which are happening, and then you could see now the PF percentage is around 65%, and the PV is 35%. So that's the current trend, the total number of cases as far as India is concerned. So it is in 2024, it is 255,000 cases, and around drastic reduction has been observed. Around 80% of cases have reduced compared to 2015 data, and also the deaths have reduced drastically. So this is the status as far as the malaria control in India is concerned, and we are hopeful that we'll be able to bring down and to the zero elimination level by 2027. Can we have the next one, please? So as far as the malaria prevalence in towns and cities are concerned, and from 19 states, the cases are predominantly, if you see the PF percentage, it is predominantly, Vivax trend is more. The Vivax is predominantly high in urban cities and towns. But then if you see the initial years, the deaths were more in 2009, around 2009, the late part of 2000. And it was mainly due to the deaths reported in Mumbai and other places where deaths due to Plasmodium vivax and leading to complications were reported and in the urban cities. But since then, cases have come down drastically. The sources again from NCVDCP, and we have data up to 2021, and then the deaths are not there as far as urban malaria is concerned. But then if you see that scenario, the cases have been drastically brought down. Next one, please. And if you categorize the high-risk towns and cities in India, so you have 3 different belts. And then if you see on the western side and the northern belt, it is basically industrial belt and the agro-based belts where you have number of towns and cities. And the cases are, as I said, on the industrial side, you have the case of, a lot of cases coming up earlier from Mumbai, and then the towns in Mangalore and other places. And then on the southern side, you have the industrial and water scarcity area, especially Tamil Nadu, Andhra Pradesh, the southern states, where you have urban malaria contributing to a lot of cases, even in Chennai. So the vector, as far as urban malaria is concerned, it's Anopheles stephensi. And the other secondary vectors also play a role, but then to a lesser extent. But then the predominant primary vector is Anopheles stephensi. And the northeastern side, you will find the industrial mining belt, where you have cities from West Bengal, Jharkhand, Odisha, and also from Agartala in northeast, you can see. But these are the towns, high-risk towns and cities as far as India is concerned. Next one. So this is the malaria prevalence in Tamil Nadu, the southern state where we are in Chennai. So if you superimpose the figure of malaria prevalence in Chennai, and on top of that, Tamil Nadu, you could see that almost the same trend is being observed. That is, the majority of the cases are from Chennai. So it is so glad to see that the cases have drastically dropped down to 3-figure in Tamil Nadu as far as the case malaria is concerned. And then also similarly in Chennai, the cases are now less than 100, it's around 133. So we are from a case status of early 90s, which was around 76,000 cases in 1993 in Chennai. It has come down to a 3-figure figure, which is successful. It's a progress which made in the state as far as and also in Chennai. Next one. Next. So coming to the city, it's it's a metropolitan city, and it's the capital of Tamil Nadu. Look, you could see the location. So we have 2 hotspot areas. One is on the northeastern side, that is, we call it as Georgetown areas, and the other one is the southeastern side where we have this Besant Nagar, Adyar, and the other areas. The population of Greater Chennai is 12.65 million. It contributes to around 15.5% of the total Tamil Nadu population. And the population density is quite high. So when we took up the study up in Besant Nagar, it was around 26,553 persons per square kilometer. So the population density was quite high compared to other cities which were, presented in American Journal of Tropical Medicine Hygiene, the comparative studies which we have done, and this was published now. So therefore, and you have high density, population density, a lot of breeding habitats, and it has got tropical wet and dry climate where summer is March to extending up to July. We are now in the peak summer period now, where the temperature is shooting up to 42 degrees Celsius, the aerial temperature. But then the things are changing due to climate change. We are getting rains now. Even the other day, there were quite a lot of rains. And then even floods are also observed during the winter season. So relative humidity being a coastal area, it is quite high. It is around 75%. And monsoon is predominantly northeast, but then when the situations have changed, we are getting frequent rains during the summer as well as the winter season, and rainfall is around 1,197 millimeter. Last few years, we are getting frequent rains. We are getting floods in December, in the odd season, so we don't know what is happening. And climate change has an impact not only on the temperature profile, but also on the different aspects, both non-communicable as well as communicable disease. And it is also playing a role for the vector breeding also. Urban malaria, as far as Chennai is concerned, and the state is concerned, Tamil Nadu is concerned, it was contributing, Chennai urban malaria was contributing to 61 to 86%. But then last year data indicates it is around 45%. The cases has come down in Chennai, therefore the urban malaria proportion has come down. Can we have the next one, please? So these are the general malaria control methods as far as the program is concerned. So you have the parasite control and the vector control. The parasite control is mainly based on the passive agencies where the symptomatic patients will approach the hospital dispensaries in both private and public sectors. And this being a notifiable disease in the tertiary level hospitals, when the cases are reported, it goes to the government machinery for recording the cases. And vector control is basically urban malaria control. It is basically as far as the larval source management, and you have source reduction methods also. The recurrent use of larvicide, the only larvicide which is being used in the program as in potable water, Stemmophos. It is, it's still, and we are finding, tolerance to, as far as the vector is concerned. And larvivorous fishes are there, and then it has been introduced in some of the wells, and most of the wells have been cleared up. But then in those wells which are still present, it is taking care of the breeding now. And you have biolarvicide in semi-polluted and polluted habitats, which being just being operated, and then aerosol space spray. You could see the photographs of the potential breeding habitats, that is over tanks, which plays a major role as far as the vector breeding. Anopheles stephensii, the vector, which is an invasive species, it breeds predominantly here. Then you have wells. The middle photo is well. And it's and most of these wells are closed now. Now you have a lot of tube wells and bore wells now. And the other one is underground tank, which we call them a sump. So in this habitat, not much breeding is there, but then these are the potential breeding habitats as far as the urban area Chennai is concerned. Next one. So we wanted to have a detailed study, a longitudinal study. We launched a longitudinal study in 2013. So we wanted to find out the breeding pattern. So we knew that our tanks, wells, and cisterns played a major role. We wanted to see whether this pattern is still continuing, or is there any preferred breeding habitat as far as the Anopheles stephensii is concerned. So when you see the table, if you see the table, weekly surveillance was made, survey was made, and then we could find out the immature density and Anopheles immature density. And similarly in wells and other breeding habitats. So other breeding habitats were underground tanks, so as I said, water storage containers, all which are contributing to the breeding of Anopheles stephensii. And this was plotted, the previous year data was plotted on the area, as well as the positive breeding habitats. And the year when we were doing the survey also was plotted. And we found that it was the positive breeding habitat was well within the reach of Anopheles stevensii, or the flying distance of the Anopheles, which is less than 600 meters. On the diagram which is shown in circular on the right-hand side, it shows the range where you find the positive breeding habitats and cases. So that shows that if there are vector breeding habitats, positive Anopheles stevensii breeding habitat, you are getting if the parasite load in the community is quite high, so you're going to get infection in that area. Next one. Next one. So we analyzed the data. We found that you could see on the other breeding habitats, we found mixed breeding as well as breeding of Anopheles stevensii. You found a varied pattern as far as other breeding habitats and wells are concerned, but you find quite different pattern as far as overtang is concerned. Around 95% of the breeding in overtang was due to the vector Anopheles stevensii. So it means that, it is the overtang water, it is irrespective of whether it rains or not, you need to have from the borewell water or from the municipal supply, you have to have water in the overtang, and that contributes to the Anopheles stevensii breeding. And it was fairly high and consistent immature breeding, irrespective of the seasons. So we correlated, as I said, we correlated with the breeding habitats, immature density, and the prevalence, and it was well within the flight range. And this indicates that targeting overtangs will control vector breeding as well as malaria in Chennai. And this has been successfully done to greater extents. That is the reason we are getting very less cases. You could see on the right-hand side the different types of tanks, the cemented tanks on top, and then the synthetic tank, plastic tanks where the sampling is made. The third one is underground sump. And the fourth one is the wells, the sampling made in open well, and sampling is being made. Next one. So we have done a couple of studies on the resting density and all, but then now since the focus is on microclimate and the climate change, so I'm bringing only the climate change microclimate studies, which we have done. So prior to this study, when we did this pilot study, everyone used to take the weather station data and plot it and correlate with the malaria prevalence, which we felt that it was not correct, because weather station is located elsewhere and then say around few kilometers, 30, 40 kilometers, and you're correlating with the ambient temperature of the vector, and which is not correct. So we did a pilot study with the data loggers, and then we found that the drastic temperature variations are there, and urban microclimates are warmer than the weather station data. And the weather station data, you will only find data either taken in the morning or sometimes both morning as well as in the evening in 5:30, whereas we don't find out the daily temperature range in this specific habitats where mosquitoes are breeding or resting. So we found that this standard weather data will not reflect the transmission condition. Therefore, working out the extrinsic incubation period, that is the incubation wherein the mosquitoes are being picked up, and the mosquitoes pick up the gametocytes, and the development takes place in the vector mosquitoes. That period also varies with the temperature which you are correlating with. So therefore, this was presented as characterizing the microclimate in Malaria Journal. And then we did a longitudinal study. The next one, please. Then you could see the, Hobo data logger, which on the left-hand side, and this was programmed in such a way hourly temperature were recorded, and it can be downloaded onto a laptop and analyzed. So this is what we have done. We found that, we could fix it up indoors and outdoors of rooftops, different roof types. It is thatched and different roof types which are there in Chennai, the tiled one, the asbestos one, the concrete one. So we kept indoors as well as outdoors, and also in outdoor resting habitats, like, for example, near to the overtang or in wells or in vegetations. We also kept that. And simultaneously, the vector breeding, the adult vector density was also being done. So you could see the bar diagram showing the vivax and falciparum cases. And as I said, vivax is the predominant malaria parasite in Chennai. And then you could see the line gram showing the man-hour density. So we could do it simultaneously. And if you see the line gram on the right-hand side, So we analyzed the data and we checked it in a day, that is in a day, in period during 24 hours, and also across the months. And if you see the top two graphs, the blue one indicates the indoor temperature and the other one indicates outdoor. So you find the indoor temperature higher in most of the time, except for the center around the noon period, where the outdoor temperature, when the sun is above our heads, and you find the outdoor temperature is quite high. But then if you see the temperature range across the months, indoor is always higher, warmer than the outdoor temperatures. So, and relative humidity is always in the outdoors it is quite high. So therefore the outdoor humidity, whether it is taken in a day or across the months, it was warm, higher, more than that found indoors. Next one. Next one, please. Yeah, and then we analyzed the data with the different the roof types, indoor as well as outdoor. You could see the table A, the graph A. So wherein the temperature was taken, and on the other side B shows the daily temperature range, the temperature fluctuations which happens in a day. So if you see in indoors, and normally not much variations are observed, but then in outdoors, the structure which is the black one shows, the line shows the over tank. The variations are quite higher in warmer temperature, but then indoor temperature, slight variations are there. But in indoor, the daily temperature range for the thatch structure is slightly more than the other structures. But then by and large, it's not not much variation. And then coming to the humidity, also the slight variations are there, but then again in outdoor temperatures, as far as the structures are concerned, so you find variations which is happening in the outdoor, in the case of vegetation as well as in structures having thatched structure. And this was used for the first time for working out the extrinsic incubation period. Otherwise, we were taking up data's temperature from the weather station and working out the weather extrinsic incubation period. We found the extrinsic incubation period for Vivax, and we worked out for falciparum also, which was varying with other structure types. And even the slight subtle changes can play a major role in the extrinsic incubation period. And that's the period when the mosquitoes, the crucial period when it becomes the mosquitoes will, the parasite development happens in the vectors, and then parasites, the vectors are ready to bite human beings. So this vector parasite extrinsic incubation period was worked out based on the real-time temperature, which we have collected from the field. And that was also published in 2018. And then next one. And then we thought, okay, we have done this data. Can we have the next one, please? Can we have the next one, please? Yeah, so this was done earlier and 10 years ago, and then we wanted to know what happened, what is happening after 10 years. And then you could see the continuous lines indicating the temperature profile of 2021, 22, that is the recent temperatures. And the dotted lines represents the temperature which were taken 10 years ago. We found that in indoor and outdoor, the asbestos temperature is much warmer than what we expected or what was there earlier. And then if you see the variations which is happening as far as the relative humidity, it doesn't matter much, but then as far as temperature, humidity will play a role in the longevity, but then temperature is the crucial part which plays in the development of the parasite. And we worked out the extrinsic incubation period. We found that the graph on the right-hand side, you can see the variation is much different as far as what we have seen in 2012 and 2013. So next one. Next one, please. So the key findings are that extrinsic incubation period is the reciprocal of parasite development, or it is inversely or negatively correlated with temperature. Therefore, when the temperature increases, parasite development takes place much faster and the extrinsic incubation period is shorter. So what we have found that significant rise in both temperature and daily temperature range from what we have observed 10 years ago. And the indoor asbestos and asbestos, which are indoor asbestos material, which is indoor and outdoor, whether it is presumed to be rust where it is presumed to rest, was much higher. So this indicates that the temperature is rising over the years. So therefore, parasite development is happening much faster, and therefore the extrinsic incubation period is much lower. So even if there's a decline in malaria cases, and the abundance also have reduced, even if the vector density is there, vectors are present there, the parasite, even if the low parasite load in the community, if it could pick up parasites from infected patients, and that could be transmitted, and it's a risk for human beings. That means it can develop faster and transmit much faster than what we thought. So this is something which is matter of concern. And then this we published last year in Parasites and Vectors. And this was also podcast by John Hopkins Malaria Research Institute, where we got a lot of comments regarding this study. And this shows that directly, and it is playing, that where is playing, climate change is playing a different role on temperature variations. And that has an impact on the vector parasite development rate in vectors. We also had done a couple of studies on the temperature, aquatic temperature, the variations, the same, the different OBO has been done. The results have been analyzed now. It is in the process of submission to a journal. Therefore, the findings of this results are not being shown here, but we have got interesting results as far as hatching rate, pupation rate, and also the adult emergence rate, because we have got DTR in synthetic tanks quite high, that is 18 degrees. And we are also getting vectors breeding in that temperature range. Next one. Next one. Yeah. So coming to the last one, the challenges as far as malaria is concerned, I've shown you the impact of climate change, temperature, humidity, and precipitation. So we are getting rains throughout the year nowadays. The temperature is increasing. Being, coastal area, humidity is quite high. So all of which favors the development of parasite vectors to develop wherever it is, though the density has come down. So that is playing a role for the transmission now, though the prevalence is low. And we have also observed the tolerance or resistance of vectors to insecticide, the only insecticide being used. So that is larvicide Termophos. And we are also working on the adult, the insecticide, which being used for the IRS. So that work is also going on. We are also finding out interesting results, the results of which has not been finalized, which will be submitted at a later stage. And what we have seen is low density submicroscopic parasitemia, which was published in Nature Scientific Reports in 2019. And we found that's, quite a high burden of, some microscopy population, and the transmission potential also it has been done by Dr. Sangamitra. So the studies have been done, and degrees have been awarded to all three brilliant, the students which I supervised. And so they have done, Dr. Shalu has done work on the aerial temperature, the resting collection over tank. Lakshmi Priya has worked on the vivax potential genetic diversity, and Sangamitra has done extensive studies on the aquatic temperature, the parasite development with the low density parasites, and this also has been published. So we are finding out different leads for it. We're finding out thermal plasticity of the primary vector. As I said, that in DTR as high as 18 degrees, we are finding the vectors to survive. So therefore, one of my PhD scholar, Kriba, is working on the heat shock proteins. So Ashwin is working on the submicroscopic parasitemia and its potential. So we also have issues related to the migrant laborers in mega construction sites. Earlier, the people who are coming for work used to stay in the same area where the construction is happening. But nowadays, people are staying elsewhere. So that is a relief as far as the vector transmission is concerned. But then things, the construction site play a major role in the, the development, in mosquito condition, especially the curing pits. And there are no proper there are no proper guidelines for contractors. Like you find rooftops and gradient is not being maintained. So after, rains, you find water stagnation. And so that, if gradient is there, water will be cleared off, but it doesn't happen there. And most of the rooftops are being used for dumping old materials, like discarded materials. And that also contributes to the vector breeding. So another thing is that due to the excessive use of personal protection measures, the repellents which are being used in indoors, where earlier when we used to get Anopheles stephensi indoors, we are not getting Anopheles stephensi much indoors. The biting also is changing. Biting pattern is changing because the first quarter of the night people are active outside, and with carbon dioxide exhalation and the sweat lure, mosquitoes are getting attracted. Outdoor transmission is also happening. So we have finding showing that positivity from outdoor mosquitoes. So therefore, it shows that outdoor biting is happening. So, and then off late water collection in the conditions and other trays, which also plays a major role in the transmission of the other vector, that is dengue Aedes dengue vectors, that is Aedes aegypti and Aedes albopictus. So a couple of my students who are working on heat shock proteins, Ashwin is working on the submicroscopic malaria. Sushmita is working on the insecticide resistant. Tanzeen is working on the midgut microflora. The phylogeny of, the phenotypic and genotypic studies on the Anopheles stephensi is being done by Madhya Rasan. And we have also a student working on zoonotic malaria. So we have more women power working on similar studies, related studies on Anopheles stephensi and malaria. And the final slide. So, we were lucky to be part of this International Center of Excellence for Malaria Research, that Center for Study of Complex Malaria in India. During the first phase of the study, a lot of studies were rolled out during that time, both epidemiological and immunological studies. I focused only on the climate-related studies. We also had, visitors like Dr. Arish Seth and Dr. Tessa discussing on the Anopheles stephensi, and which was published in 2023. And we were also involved in the global framework for the response to malaria in urban areas, and which was published by UN, both UN and WHO. And I would like to acknowledge ICMR, National Institute of Malaria Research, the former directors and the directors, director of NIMR, and also the collaborators of ICMR. Many of them had helped us in getting all the things in the right time. So, and also the program officials regional office, where we did our majority of the studies in Basanagar, the regional directors and the staff there, the state program officers, and of course the Greater Chennai Corporation health staff. And I also thank my staff and my team who had helped us and working out for doing a sincere and dedicated study. Thank you so much.
Excellent. Thank you so much, Dr. Alex. That's a hugely impressive piece of work. And it's just really impressive to see that data and also that importance of the local infrastructure and what's on the ground with determining mosquito transmission. So again, just to remind everyone, I see a few questions coming up, but please do add any questions to the Q&A box. And we will have a session for all question and answers later on. So with that, I'd like to introduce our next speaker, Dr. Stefan Karl. So Dr. Karl has a master's in biological process engineering and a PhD in biophysics. He has done a lot of work in vector-borne disease epidemiology and entomology, and is currently a senior research officer at the Australian Institute of Tropical Health and Medicine at James Cook University, as well as the laboratory head of the entomology section at the Papua New Guinea Institute of Medical Research, where I believe he is joining us from today. So Dr. Stefan, I'll hand over to you now. Thank you again for joining.
Just confirming that you can see my slides.
Yep, all good. Thank you.
Okay, thank you. So let me go ahead. Thank you so much for the opportunity to give a presentation here at the meeting today. I would like to talk about climate change and temperature suitability for malaria transmission in Papua New Guinea. I'm trying to advance my slides, not working right now. Oh yeah, here we go. Okay, Papua New Guinea reports about 1.2 million malaria cases per year. That is 85% of all the cases in the WHO Western Pacific region. However, treatment seeking is only estimated at 40%. So you can imagine that the true number of cases in Papua New Guinea is much higher than the 1.2 million that are being reported. About 40% of PNG's population live in areas above 1600 meters, where temperatures are too low to support a stable malaria transmission. And you can sort of see this here in the maps, the elevation map on the left-hand side showing the high mountains in Papua New Guinea, lots of areas above 1600 meters. And the population density map on the other side of the screen overlapping with these highlands regions. I have indicated in this population density map the areas above 1600 meters where about 40% of the entire country's population lives. So this analysis explored if rising temperatures along with climate change, will shift transmission of malaria to higher altitudes in the future up to 2040. The aim was to build an understanding about the impacts of climate change on malaria transmission in PNG to inform National Malaria Control Program preparedness. So you can see again in PNG, like everywhere else, the temperature is dependent on the altitude, so it's much colder up in the mountains, and that is where a lot of the population is concentrated. So I have to go a little bit into mathematics, but I promise it won't be too much. Many mosquito and parasite parameters that govern malaria transmission are functions of temperature, and we heard a little bit about that in the previous presentation. So biting rate, vector competence, things like mosquito and parasite survival, development rates of parasites and mosquitoes, etc. These are all functions of temperature. And so you can combine all of these different parameters, and people have studied them, right? They have studied the temperature dependence of these different parameters. And so you can combine all of these parameters into a quantity called the relative basic reproduction number, or R0, which then in itself also becomes a function of temperature. And you can use that to sort of illustrate the suitability of a transmission environment for malaria transmission. So it becomes this horrible formula that is plotted here, but you don't need to think about that very much. You just need to remember that malaria, the suitability of an area for malaria transmission is a function of temperature, because all of these different mosquito and parasite parameters underlying transmission are in themselves function of temperature. So smart people have done this and have done meta-analysis looking at what that actually looks like and fitted that. And so this is at the curve of the basic reproduction number as a function of temperature based on data. So based on this, malaria transmission is possible between around 18 degrees and 34 degrees Celsius with an optimum of around 25 degrees, right? So this is a relative suitability, temperature suitability, as the absolute value obviously depends on other climatic and socioeconomic factors. So you can think about it in this way, that temperature within this range is necessary, but obviously not sufficient for malaria transmission. You need other things. You need humans to be there, you need mosquitoes to be there, you need habitats, etc. So so it is a necessary but not sufficient prerequisite for malaria transmission. And so the question was how this relates to altitude in PNG in the past, present, and future. And I'm gonna, this is a shortened version of this presentation. I'm only gonna focus on the present, the recent past, and the future. So a little bit more methods. So I got the monthly temperatures from WorldClim for the decade 2010 to 2019. I got a future climate prediction for the period of 2021 to 2040 based on these shared socioeconomic pathway models, also from WorldClim, and the general circulation model developed in Australia. I've got lots of data from Papua New Guinea, administrative boundary, digital elevation map, population density distribution at relatively high spatial resolution. Took all this data, overlaid it with a 2.5 by 2.5 kilometer grid covering PNG. And then from the temperature data, you can calculate this relative basic reproduction number that indicates the degree of temperature suitability for each of these grid cells. And so you can basically map this relative R0 across PNG. For the more visual people, here's another slide just showing the different data sets that went into this. I'm not going to talk about malaria incidents in this presentation, but temperature obviously went into this analysis, the administrative boundaries, altitude, population density, and temperature derived the basic reproduction number in a spatially explicit format, as you can see here. And so all of this applied to this 2.5 by 2.5 kilometer wide grid spanning PNG as illustrated here. A few words about these future climate predictions based on these shared socioeconomic pathway models. So, so these are models that assume how the global society sort of like works in the future, and they can be more optimistic or more pessimistic depending on depending on these broad-ranging assumptions that they are making. So I chose a relatively conservative one, which is called SSP 245. It's called the middle of the road scenario that basically extrapolates the current situation into the future. Based on this scenario, we're expecting something like 3 to 4 degrees Celsius increase in global temperature by 2100. So that's a relatively conservative scenario because we don't want to do a lot of fear mongering either. There's certainly much more pessimistic scenarios as well, as you can see here on the slide. So when we compare the temperatures based on these models between the decade of 2010 to 2019 and 2021 to 2040, then we can see that we are expecting an average temperature increase of around 1.3 degrees Celsius across PNG. So between now and 2040, the average temperature in PNG is expected to increase by about 1.3 degrees Celsius. And that, as you can see, sort of like on the top 2 panels, a map of the temperature increase on the left-hand side and a graph of it for each grid cell on the right-hand side. And that you can then translate using that horrible formula that I showed on one of the previous slides into this basic, into the change in the basic reproduction number between these decades, 2010 to 2019 and 2021 to 2040. So you can see that delta R0, the change in the basic reproduction number, is quite significant. In particular for regions around about 1,000 to 1,500 meters in altitude. You can see this on the map. It's this sort of heat map where the highlands areas are regions where delta R0 increases the most. Or also you can plot the delta R0 over altitude, and it just illustrates the same thing in a slightly different way, showing that the change in temperature suitability for malaria transmission is greatest in an area of about 1,000 to 1,500 meters, but also above that. Coastal areas are actually predicted to decrease in their temperature suitability. It becomes too hot, but since the temperature range in these areas allows for an R0 of close to 1, so optimal transmission conditions, we wouldn't expect this decrease to result in a massive decrease in the suitability for malaria transmission in these areas. So, and because it's all very nice and spatially explicit, you can then also look at various locations specifically here. So you can do these kinds of case studies, as I call them. I just have one example here, a town called Goroka in Eastern Highlands province in Papua New Guinea at 1,500 meters roughly. The population density map shows sort of the location of Goroka there. This high population density area in the northwest of the province, indicated with the red circle. The delta R0 map, so the change in the temperature suitability between 2010 to 2019 and 2021 to 2040 in the center panel there. And then the difference illustrated, the difference in absolute R0 values illustrated in the rightmost panel there. So you can see that R0 increases from something like, and it's shown in terms of seasonality as well. So you can see that R0 increases quite substantially between 2019 to 2040, and the place becomes much more suitable for malaria transmission. So what can we say about this? Within the present and next decade, until around 2040, temperature in PNG is predicted to rise by about 1.3 degrees Celsius or Kelvin, it's the same thing, in a relatively conservative climate change scenario. And this will lead to a shift in the altitude range, sustaining malaria transmission by about 260 meters. And as such, malaria will become more endemic in highlands areas of PNG that are currently not malaria free, but that currently don't sustain transmission at significant levels. And more people will live in areas that actually become malaria endemic. So the question is, and now we are going into sort of like, how this kind of research can inform preparedness of national malaria control programs. What does that mean for the total population, for example, living at risk for malaria transmission? And in my last slide. So now we're looking at past and future population at risk estimates. These are not case numbers, and malaria case numbers or something. This is purely the number of people living in malaria endemic areas of PNG. So in 2019, PNG had around 11 million people in total. Of these, about 6.5 million lived in malaria endemic areas, so below 1,600 meters. And now I've got two models. One model includes population growth only. So temperature increase wouldn't be happening. It is assuming the same kind of average temperature continuing until 2040. And then I have another model that also includes this temperature increase. So by 2040, we will have a total population, expected total population in PNG of about 18.5 million people. And the population growth only model would predict that about 11 of these 18 million would be living in malaria endemic areas. Those would be all the areas below 1,600 meters again, roughly. If we take the temperature increase into account and as such the increased area that becomes suitable for malaria transmission, the total population living in malaria endemic areas would increase to something like 14 million. So that's a difference of about 3 million. Okay, so temperature increase, climate change will make a difference in terms of how many people the program would have to be prepared to supply with preventative measures, treatment, diagnostic facilities, etc. And I've just plotted it here again for these different scenarios also over altitude. So in summary, the total population living in risk areas of malaria transmission in PNG is expected to roughly double between now and 2040. And 50% of that increase can be expected to be due to the rising temperatures. So maintaining the current levels of malaria control in Papua New Guinea will require substantially more resources. So we're not talking about making much progress towards malaria elimination or something like that, but we are talking about maintaining the current status quo, because the number of people requiring tools, measures to protect them from malaria, let's say insecticide-treated nets, etc., will increase substantially. And if you're looking at the global climate in terms of funding for malaria control, for example, what's happening globally in this sector, that does not bode very well. And I think it's important for national programs to consider these kinds of things. And this is just kind of like our little contribution to that sort of preparedness. Thank you very much.
Excellent. Thank you so much, Dr. Carl. That was really a fantastic overview of your work in Papua New Guinea, but I think also really important to show actually the impacts that has and how that can be used to plan control, or thinking even about what resources are needed for control. So again, I think now we'll kind of move into our Q&A session. So if all the speakers could turn their cameras on. And if anyone has any further questions, please do add them to the Q&A box. We'll try and get to as many as possible. Excellent. And Dr. Alex, I don't know if you're able to turn your camera on. But I will start off actually with a question for Dr. Tung. And that is, so you've been very clear and you've answered some of the questions already that you're specifically looking at climate hazards, but there's a question about how we can associate these compound hazards with their impacts. So for example, mortality or morbidity from the population. And I actually haven't seen many examples from this in the literature, so I'm very curious to hear your thoughts.
So actually, we do not have an exact number on that. So, but if we consider the occurrence, so the occurrence of the single hazard are much more frequent than the compound hazard. So, personally, I think that the number of people affected by a single hazard will be much higher than a number of people affected by the compound hazard. However, the impact of the compound hazard may be more severe because from the first hazard, people suffer, and then they do not have time to recover from the first hazard. And then when a second hazard occurs, they will be more, exposure to the more exposed to the hazard, and the impact will be larger than the sum of just a single hazard that occur individually between each other, like that.
Yeah, excellent. I do completely agree. And I think it's really important to think about. So we've certainly seen the examples from Pakistan where there's kind of major natural disasters and multiple hazards that occurred at once, and those had really kind of substantial impacts on malaria transmission. And so thank you again. So my next question will be for Dr. Alex.
Yes.
Let me just see. I lost it. So there is a question about what do you think was the impact of surveillance? So we're for urban malaria, is it much more likely to be detected because it's in an urban area versus a rural area? And there was another question about how that relates to other mosquito-borne diseases such as dengue and arboviruses.
Yeah, in urban areas, the interventions are basically larval source management. So we could easily identify the breeding habitats of Anopheles stevensii. But then the resting of these mosquitoes, vector mosquitoes, it rests in many cryptic places where it is really difficult. Of course, in cattle shed, cow sheds, and in those habitats, in dawn and dusk collection could help you out to get these vectors. And then we give this data to provide this information to the Greater Chennai Corporation, who in turn will take intervention measures. But the major factor is that as far as over tank is concerned, is the type of tank which has been used now, the synthetic plastic tanks, which were earlier a flap type of lid, which could, if it is not closed properly, because of the frequent, wind action being a coastal area, it could open and then close, and the frequent flip-flap, closure, and also the exposure to the sunlight and then rains, it could brittle, and then you could see the lids not in proper shape or broken. And this, what we have advised is, and to a couple of the company people, were to have a screw type lid, so which could screw it up, and then the flip flap movement was. So in most of these tanks, so now we have a proper lid. So, and that has been taken care as far as the vector is concerned. And the number of wells also have come down, because many people now, the supply is much better than the earlier days. And the housing types have also changed. Wherever, when we did the study initially 10 years ago, the longitudinal study, we found a lot of thatch structures, and that is slowly changing to asbestos roof type, where you find the resting of the vectors are less. So the combined effort of the interventions made by the health department of the Greater Chennai Corporation, and the support by the research findings from the scientific institute like ICMR, like us, used to be helped them to get rid of these vector breeding as well as the infections. And that is one thing. And then in our study also, we did community-based epidemiological survey to find out the case, and then that was shared with the program. And then even in low density cases, treatment was given. So that is something which is appreciated, because in the program, so it is either by conventional microscopy or RDT treatments are given. So we need to think about, if the low density malaria parasite population is there, so it is there in the community, what action has to be done. So this is also something which is matter of concern now. So the effort from both the program as well as the research organization has helped in bringing down the cases, and it has drastically come down, and compared to a few years ago. Yes.
Fantastic. No, thank you so much. And I think it's really impressive, the work that's been done, and really identifying those kind of key priorities for future surveillance.
Thank you.
My next question for Dr. Stefan is around El Nino. So Jeffrey has asked, how strong is the evidence of El Nino, and how does this play a role in malaria outbreaks in the 5 highland provinces? So he mentions as well, there is an example in the neighboring Papua province, when drought conditions and elevated ambient temperature and population movements led to falciparum epidemics.
I sincerely apologize, but the internet just dropped out right when you asked your question, and I have not heard it. Something with
No
El
problem.
Nino, and then it cut off.
That usually, yeah, it's always the timing. So the question was, how strong is the evidence of El Nino playing a role in malaria outbreaks in the highland provinces? And I think there was some historical examples from the 90s in Papua mentioned as well.
Yeah, there is some evidence for this, but I think, yeah, we have not looked into this in great detail, and I can't actually really speak much to this. So there is outbreaks in the highlands. So that's a kind of like a malaria outbreak scenario, kind of indicating that there is a certain transmission window there, but it's probably very short, depending on what altitude you're at. But yeah, we don't have very good information, I think, about, you know, I would hypothesize that many of these outbreaks are not detected in these kinds of locations, for example, so we don't have information coming in. So I can't actually really respond much to this question because I simply don't know.
No, no problem. There's always more research to do, so I think it's quite useful to highlight. I'll kind of merge a few questions together, but actually I think it kind of comes to this point. So this is a question for everyone, but we've seen a lot of different examples of different kind of temporal and spatial scales of climate data used. So looking at long-term impacts versus looking at very, very fine scale impacts. And so what are some of the ways that you think we could link some of these together? So for example, linking some of these larger changes in temperature to microclimate and kind of thinking about how we could potentially design landscapes to address some of these challenges. So thoughts from anyone? I guess maybe I'll start with you, Dr. Stefan, just because the question was originally written, they were asking about the role of microclimate in the types of models that you've done. So whether kind of you find it's similar how you could incorporate some of the work that was done in India or similar methods in the future into these types of predictions.
I think they would have to be included. So they would clearly have to be included. Like, what we have done here is a very simplistic approach, looking at you know, only one parameter, which is temperature. But we know that malaria is obviously determined by many other factors, rainfall, land use, etc. And I think there is a lot of data, satellite-based data available. Some data you will not be able to derive from satellite imagery. So we're doing a lot of vector surveillance, for example, on the ground, trying to increase our knowledge about the vectors that are actually present in the highlands and how that might change. So, so yeah, I think there's a lot of scope to combine all these different data sets, and you need these different data sets to develop better models of what's going on. The question in my head is always a little bit like, you know, how detailed do these models need to be in order to inform programs? Okay, so I think there's a certain pragmatic, you know, sort of pragmatic, let's say, a threshold where the model just becomes maybe too detailed, unnecessarily detailed, and the added detail doesn't really add anything to your ability to control malaria.
Yeah, I think that's a very fair point, and it does depend on what the question is sometimes, or how the data is going to be used. Do you have any thoughts, Dr. Alex? So I know you've used some wider climate change projections alongside the microclimate models and kind of monitoring.
Yeah, we have used the microclimate data for the parasite development and working out the extrinsic incubation period. But then we need to have proper data set, real-time ground level data set to find out these variations which are happening, because we can't, you know, climate change is happening and then, you know, you find then you find the next morning, rains are there during winter season or during summer season, and things are going to happen. So that plays a major role. So unless you have this proper data set, with that only you can work out this better model and then validate it whether it's working out. If the data sets are not, you know, up to the mark, then if it is in a macro level, if it doesn't work out at a macro level, then it may not give the right predictions. That's why whatever predictions are given sometimes varies unless you have, you know, proper, you know, ground level data sets to find out. So rather than doing, you know, macro level things, yeah.
Yeah, no, I definitely agree. I think both sides of the story are really important, and there are things you can measure at scale, and there are things you can measure on the ground, and it really needs the input from both. So thank you both for sharing your work on that topic, because I think that they're really complementary. We're almost out of time, so I'm going to end with a last question for everyone from Indra. So of course it's difficult, but for everyone kind of just to think, Indra has said, climate change has been gradual and many countries have reduced malaria cases over the years. So what is next? So kind of what are your key thoughts on how we can work together, how we can kind of address some of the changes of climate change for both malaria or even other diseases that are driven by climate change? Dr. Tung, could I call on you to go first? I know you've kind of moved from climate science to now working more with dengue. So what are your thoughts on some of the opportunities here?
So personally, I think that the climate change uh, we need to some challenge because we may have an increase in the high temperature event, and also in some area we may also have an increase of the strong precipitation events, and this strong precipitation event may lead to the increase the chance or possibility of the increase in the how to say the breeding size of the mosquito, and then may lead to the we need to the larger effort to manage and to reduce uh, uh, the impact of the mosquito on health. Yeah, I think yeah.
Excellent, thanks very much. And I think these partnerships between climate science and some of the epidemiological work are really quite critical as well.
Yeah.
So Dr. Alex, any thoughts from you? Climate change and malaria?
Yeah, well, climate change and malaria we've been discussing, but then other vectors you were telling about dengue and all. So intermittent rains will lead to breeding of dengue vectors, especially in peridomestic areas where you have lot of breeding habitats in discarded and other habitats where you get number of breeding habitats. Even the collection of water can lead to the breeding of Aedes aegypti and also Aedes albopictus, depending on the vegetation. So earlier it was mainly on the water storage containers. So now it is more than water storage containers. Now, as far as dengue is concerned, you have peridomestic areas with intermittent rains, and then the temperature playing a role for the breeding of these vectors, and then resting elsewhere. So I mean, that is something which, as far as dengue is concerned, and even when we had flood in 2015, after that large water sheets collections, not large water sheet collections were observed, and we could find vectors of GE, also Japanese encephalitis, also breeding in railway yards and places. So therefore, it's not only for malaria, but then for other vector mosquitoes also, it can play a role in the abundance, the density, and ultimately, if the parasite or viral load is there, then it could transmit infections much faster. And especially Aedes is it's much dangerous because it's a multiple feeder. We call them a SIP feeder. So therefore, though we say the biting rate is a reciprocal of gonotropic cycle duration, but then it is since this has got a multiple biting phenomena, so that the risk is more as far as Aedes is concerned, or dengue vectors are concerned. So therefore, the climate change with the intermittent rains and will play a major role as far as the vector is concerned. And then if you have flood and after post-monsoon, you have other mosquitoes breeding in that. And depending on the type of water quality, like, the fresh water, the clear water, you find breeding of other anophelines like Subictus vagus and all, Anopheles subictus, Anopheles vagus, and other vectors in, and then you write once it becomes and you find Aedes aegypti also breeding in along with that in small containers. And then later stage, it becomes polluted. You find Culex conguifiscitus and other vectors may. So therefore, water stagnation is one thing which is there. If it is not stagnating beyond a week or so, then slowly the percolation happens in the earth. So then the soil takes it up, then the issue is not there. But then in other habitats, of course, as far as dengue is concerned, it's a major concern, major issue. Yeah.
Excellent. It is really clear there's still a lot of work to do, and kind of a lot to make sure that we don't, we continue to maintain the gains that we've achieved in malaria control and moving towards elimination. And so, Dr. Stefan, any kind of thoughts about what's next steps?
Well, I think that, malaria control programs need to take climate change seriously and into account in their planning. So like my presentation was only a very simplistic sort of like scenario of, what might happen with temperature increase, but obviously it's more complex than that. So I think programs need to define and measure, key indicators of climate change somehow, and use these sorts of forward projections to enable them to prepare and plan for the things that might happen. I think that would be useful. And so I think there's a lot of opportunity there for multi-sectoral collaboration and multidisciplinary collaboration between researchers and program people and different disciplines of research to enable that in a better way.
Excellent. Well, I think that's a perfect statement to end on, because I obviously very clearly agree with that. But I do think it is really important to be very aware of these changes and to start to think about how we design surveillance systems that can detect these changes at different scales. So some of these longer term projections, some of the factors that are really influencing these fine scale spatial and temporal distributions, thinking about where we have multiple risks occurring at once and how that impacts transmission. So I would just like to thank you all again for sharing your work. I think it's really important to try and build this dialogue. So we're very grateful for you for spending your time. And thank you to everyone who joined. And we hope to see you for the next webinar. So you may see a poll come up. So it's really helpful for us if you actually could give us some feedback, and we'll try and make sure we can continue to develop these webinars in a way that's helpful for you. So thank you again to everyone.
Thank you so much, Dr. Kimberly.
Thank you very much.
Thank you very much for organizing this webinar.
Thanks for weeks.
Thank you.